Hello, and thank you for joining me for today's photosynthesis webinar. I'm Louisa Diva, and these webinars are sponsored by the Capitalise project. Today, I'm very pleased to welcome our two guest speakers, Tom Theeuwen and Marc Heuermann. I'm going to hand over to Tom, who's going to talk to us. Tom is a postdoc at the Laboratory of Genetics at Wageningen Plant Research, and he uses high throughput phenotyping in dynamic environment conditions to reveal physiological properties and genetics to determine their underlying mechanisms. And today, he's going to talk to us about natural variation and how it can be used for crop improvement. Over to you, Tom.
Thank you very much, Louisa. Indeed, in the next few minutes, I would love to give you a short overview of the things that I've been doing in the PhD that I just finished. And my PhD was entitled Exploring the Unexplored, Unraveling the Cytonuclear Interactions in Arabidopsis thaliana to Improve Photosynthesis. So first off, some of you might be thinking photosynthesis and Arabidopsis thaliana. Why are we still studying in Arabidopsis thaliana? And I show this with a very few simple images. This is Arabidopsis growing in the Netherlands in spring. And most people living in Western Europe will recognize this as the natural habitat of Arabidopsis. But these are also two pictures, just two examples. I could have used many more that are also habitats that Arabidopsis is native to. And as you can probably appreciate by the differences in these pictures and the locations given, there's a huge diversity in the environments between those, but also between those and for instance, Western European countries. And whenever there's environmental variation, there's room for adaptation. And there's good evidence that also for photosynthesis, adaptation to these environments has occurred. And it's my personal motivation to use Arabidopsis thaliana to actually study this variation for photosynthesis and see how we can exploit that. So, being a geneticist, I want to know where does the genetic variation manifest itself. So when we look at a very simple drawing of a plant cell, we have a nucleus, a mitochondria, and a chloroplast. And if we zoom into these three compartments in the case of Arabidopsis, we can see that the nucleus has roughly 27,000 genes, while the mitochondria has only 90, and the chloroplast has roughly 120 genes. We all know that photosynthesis primarily takes place inside the chloroplast, but it also uses the products of roughly 3,000 genes to actually accomplish this. That means that in order for photosynthesis to happen properly, a lot of proteins have to be coming from the nucleus and be transported to the chloroplast. So there's a very high directional flow in that sense. However, in order to make the process go well, there's also the products of the gene in the chloroplast that are actually giving feedbacks and thereby establishing whether a plant can do photosynthesis as well. And this is a cytonuclear interaction. And it's my aim to actually study how variation for the cytonuclear interaction influences photosynthesis. So how can we study this cytonuclear interaction? Well, I do this by actually unraveling it or disentangling. And what I call, so the genomes of both the mitochondria and the chloroplast, I together collectively call the plasmatype of a plant. And what I would love to do is to be able to swap plasmatypes between accessions of Arabidopsis thaliana. And it would look something like this, take them out and put them into the other one. And the resulting genotypes I refer to as hybrids. And these hybrids would allow me to study this cytonuclear interaction. So in practice, how do we make these hybrids? Well, I use for this a so-called haploid inducer line. And I use for this a very simple crossing scheme where I have a haploid inducer line as a female and a wild type accession as a male. And these two stripes here, they represent the nuclear genome, and the circle around it represents the plasmatype of these plants. This haploid inducer has something specific in that it has a mutation in the SNH3 gene. Normally, when you cross two plants, the offspring will have 50% of the nucleus of the mother, 50% of the nucleus of the father, and the cytoplasm of the mother. But due to this SNH3 mutation, the maternal genome is lost after a few cell divisions. And as a result, we have a haploid, and this haploid I can make into a double haploid. And this double haploid has the nuclear composition of the father and the plasmatype of the mother. And this is effectively a hybrid. And using this simple, relatively simple approach, I could give the plasmatype of one accession into 7 different nucleotypes and all the other combinations, creating a so-called hybrid panel. This was already a proof of principle that this is actually possible, but I was not in the business of making hybrid panels. I was in the business of using hybrid panels to see whether there's natural variation for photosynthesis. So the hybrid panel was ready, but was it useful? To actually study this, I teamed up with Professor David Kramer from Michigan State University, and he has so-called DEPI systems where we can use very dynamic environmental conditions such as light and temperature, and use a range of chlorophyll fluorescence imaging systems to actually monitor them all at the same time. And in this system, we can expose these plants to a range of different environmental conditions, in this case, light intensity, and then see how these plants behave. And when we look at the lower light conditions, so this is only 200 micromoles per square meter per second, for a range of phenotypes, photosynthetic phenotypes, we could see that the heritability was up to 80%, which is very good. However, the plasmatype of this was actually contributing only to 3% of the total variation. And this is actually a bit disappointing. However, when we were exposing these plants to sinusoidal light, so higher than they were used to, and especially under fluctuating light conditions, some of these photostatic parameters are actually contributing much more due to variation in the plasmatype. And this is very, very interesting because this is variation that we normally never are able to pick up on. In some cases, 1/5 of the variation was explained by variation in the organelle genomes. So being very excited about this, I zoomed into what was causing this variation. And here I show the averages of the 7 plasmatypes for 5PS2 and for QE. And what you quickly can see is that there is one plasmatype, the so-called Ely plasmatype, that has a severe reduction in 5PS2 and a severe reduction in QE, the fast reaction component of MPQ. But this was not such a surprise. I included this because it has a known mutation in the PSBA gene, and it was there as a sort of control. However, what we did see is there was one accession that actually had slightly higher 5PS2, while also still having severely reduced QE. And this was an effect that was completely unknown to us before, showing that there is actually quite a little bit, or there can be quite a bit hidden variation within the organelles that we can actually use to, first of all, study photosynthesis, but also use it to improve photosynthesis. So this was a very nice proof of principle. But roughly around that time, in 2016, 2017, the genomes of quite a lot of Arabidopsis accessions came available. And together with some bioinformaticians, I reassembled these focusing on the chloroplast genome, and I could have made a phylogenetic tree of the Arabidopsis thaliana species. And that is represented here by this tree. And the 7 accessions from this first hybrid panel were, to my surprise, quite nicely distributed around this tree, around the branches of this tree. However, you can appreciate that there's quite some branches that are still not being represented in this panel. So potentially I'm missing a lot of variation within this hybrid panel. So to actually get a good overview of the species-wide variation within Arabidopsis thaliana, I generated a second hybrid panel where I combined 60 plasmatypes, the ones here shown in yellow, and I combined it with 4 highly divergent nuclear genomes. So using this big, this was a lot of work, that once I had it available, I was actually interested in studying dynamic photosynthesis. And as you probably are aware of, the light, sunlight is rarely stable. And as a result, photosynthesis responds to it, but it tends to be relatively slow. And what we would love to is photosynthesis to be much more in tune with the light environments. So what better way to mimic outside conditions than to actually go outside? And that's what I did. I grew this hybrid panel in 2 consecutive springs in 2020 and 2021 here in Wageningen. And at the end of the growing cycle, just before they started flowering, I took them all inside and I phenotyped them using a high throughput phenotyping system based on chlorophyll fluorescence imaging. And I expose these plants to a short light regime, low light, high light, low light, and I monitored the response, in this case, 5ps2. So the faster a plant could recover after a highlight treatment, the better it recovers to this kind of thing. So this was a trait that I was looking to see whether there was variation for. And when I take all of the 60 plasma types in combination with one nucleotide, I could see 2 plants that were actually recovering 5ps2 faster than the other ones. And to my surprise, this was the Burr plasma type that we already witnessed in the earlier panel as well. And when I did this for all of the nucleotypes, so across all nucleotypes, I could see that very persistently this Burr plasma type had a higher recovery of 5ps2 regardless of the nucleotype. So I already had seen it before in inside conditions, but now also outside, I saw there was variation in this Burr plasma type. So, and now it was actually recovery of 5ps2. That was actually very interesting to me. So I zoomed in a little bit more. I grew these plants in such a way that I could monitor how these plants responded from high light to low light. And you can see here the response of Colombia, so that would be the control. And we would love to have a response that is as quickly as possible as depicted here in this hypothetical red line. The Burr plasma type, however, even though it started slightly lower in terms of 5ps2, it very quickly got higher, and throughout the whole phase, it was significantly higher. So there's really something to this Burr plasma type that actually is able to recover 5ps2 faster. And being a geneticist, I actually want to know what is causing this. So there's ample of genetic approaches to find causal genes in the nuclear genomes, but this is in the organelle genomes. So that's actually much more complicated. So for this, I developed a so-called genetic exclusion approach. And to cut a long story short, after a lot of rounds of genetic exclusion, I was able to find that allelic variation in so-called NDHG gene that actually was causing this. And the NDHG gene is a subunit of the NDH complex. And the NDH complex, what is the function of that? Well, we probably are familiar with the linear electron transport of photosynthesis, where in very short, it produces NADPH and ATP in a certain ratio. But besides this linear electron transport, there's also cyclic electron transport. And in this case, the energy of ferredoxin that normally is destined to go for production of NADPH is shuttled back into the so-called NDH complex here in blue, and the electron is being used to pump proteins over the membrane. And in this way, the NDH complex has influence on the ratio of NADPH versus ATP ratio. And the one that I had a allelic variation for between the 2 plasma type was one of the proton pumps, the NDHG. So that was also a very nice, I could form very nice hypothesis on this, because like, what is the influence on NDH activity? There's actually a very nice way of measuring NDH activity using chlorophyll fluorescence. And I did so using a knockout of one of the NDH subunits that showed very nicely to have no NDH activity. Then I used the Burr plasma type and the Colombia plasma type as a comparison in 4 different nuclear backgrounds. And while you can see that there is variation for the nucleotide, the Burr plasma type every time has a significant reduction in NDH activity. Very important is that it's not a knockout of it. So it's reducing the NDH activity. And this reduction in NDH activity coincides with the fact that the Burr plasma type has lower MPQ, QE, in lower light conditions, which is actually a very desirable trait when you're improving photosynthesis. So far, what I show you is that while the NDH complex, which is known to be very conserved among higher plants, there are some exceptions, but generally it's considered very conserved. We have a very rare variant in NDHG that can be used to improve the efficiency of photosystem 2. Taking from this is that the NDH complex probably plays a larger role than previously considered, because in Arabidopsis thaliana, the knockout NDH did not show any phenotype. So what I'm now doing is using this variation in the NDHG gene to study the role of the NDH complex in much more detail. But there is more to this. I showed you that I made a cyber panel having 60 plasma types, and I've only zoomed into the effect of only one plasma type, which is this Burr effect. And I've grown this cyber panel in 2 different high throughput phenotyping systems in a range of environmental conditions, both dynamic light and dynamic temperatures. And these principal component analyses show a nice overview of this. And while we already could see that the Burr effect was quite different, it's nowhere near the most extreme ones in this. So that tells me that there is actually much more variation within the organelle genomes that we are yet to tap into, learn from, and use to improve photosynthesis. But still, I told you, the plasma types only make up a very small fraction of the total number of genes. So what about the nuclear genetic variation? So again, I come back to the cyber panel that I showed you before. I have again the 7 plasma types that I showed you in the very beginning, but this time I show how they respond in combination with a nucleotide. And what you can see that 5PS2 again is reduced for the ones that have the ELI plasma type. And when you do the same for MPQ, you see the same. However, surprisingly, I saw that the ELI nucleotype, so the one that is in the wild occurring together with the ELI plasma type, has a higher MPQ. And this was primarily due to a difference in QE, and that could potentially be a compensatory mechanism to this reduction in 5PS2. So I got very interested in how a plant could adapt to such extreme differences in 5PS2, and potentially I could use this difference in QE to improve photosynthesis. So I zoomed in, I exposed these plants to a conversion from high light to low light. And I could see, even though the MPQ was in the beginning a little bit higher, in the first 100 seconds, the MPQ was significantly lower than the Columbia control. And while most of this, so there has been quite a bit of work going into this kind of phenomena, and mostly this is associated with variation in expression of the VDE, ZEP, or PSBS genes, proteins. But in my case, between these 2 genotypes, there is no genetic variation to be found between them. So that must mean it's being caused by something that is not really on the radar of us yet. So for being a geneticist, I wanted to reveal this, and so I created between these two parental lines, I generated a double haploid population of 500 lines, and I used whole genome sequencing to actually be able to do QTL mapping on this. Again, I did it in collaboration with the lab of David Kramer, and we grew these plants to a range of different light intensities, and I started doing QTL mapping. So at very low light conditions, I did a QTL map, and here's the results, the 5 chromosomes of Arabidopsis, and you could see it like 1, 2, 3, 4, 5 places. There's a significant QTL, there's a QTL associated with MPQ. And when I plot this, you can see these 4/5 QTLs displayed here. And when I do this for the whole dynamic environment conditions, you start to appreciate how many QTLs they're actually located, or actually segregating between 2 parental lines. So while there were 4 QTLs in the beginning, some of them disappear at the end of the day, some of them disappear only after a day, but there's also QTLs that come that appear that were previously not there. And moreover, you can see that the QTLs that are depicted in green are caused by an allele by the ELI parental line to have higher MPQ, while the Columbia allele is depicted in blue if it's higher. So this means that there's 2 genotypes that have completely different ways of doing MPQ using their own natural genetic variation. So I'm now focusing on this specific QTL, which if you zoom in, has a difference in roughly 35% in QE. And this might actually be explaining how these plants are able to, or how ELI is able to recover from MPQ much faster, or relax MPQ much faster. So that brings me to the end of my talk. What did the unexplored reveal? Well, I think it's safe to say that there's ample of genetic variation for photosynthesis. It's located not only in a nucleoside, but it's also located on the organelle genomes. All of this is a result of a highly complex genetic architecture. So you could see that within the nucleus, it's segregating for a lot of different QTLs. And therefore, due to this highly complex genetic architecture, I am strongly advocating the fact that we should first improve photosynthesis before we can improve yield. And moreover, I'm very convinced that the fact that we have this natural genetic variation, we can actually use it to study photosynthetic functioning in more detail. And with that, I come to the acknowledgements. I want to thank my promoters and co-promoters and my collaborators during my PhD project. Are there any questions?
That's what I wanted to say. Thank you, Tom. That was very nice. So please put your questions in the question box and I will kick off the questions. So how easy, I mean, I'm presuming this hasn't been done in any crop plants before. It's not a method that people use for crop breeding at the moment. Would that be right?
No. This is definitely not done in crops before.
Because in theory, it'd be a nice way, I presume, to take something distantly related, like a crop wild relative and an elite line, to be able to harness some of that variation. Is that something? How easy would it be to transfer what you're doing towards crop improvement?
That's an excellent question. This method that we are using now in Arabidopsis thaliana is until recently only available for Arabidopsis thaliana, and it limits you to do crosses within species that are able to be crossed with each other. So if you are able to cross these distant relatives amongst each other, that might be useful. But it relies heavily on this SANH3 mutation, which is only so far working in Arabidopsis, and some exceptions. So in some species, it's also possible. But it's a very hot topic in science, and I presume within a year or 2-3, this is available in a range of crops. Though what I should say with this, it's not the only way to make these hybrids, because by recurrent backcrossing, you can actually achieve similar genotypes, though being it a longer process and a little bit less precise, but you can achieve doing this. So it is accessible for crops already now, and it was already available.
Okay, thank you. I have a question here, congratulating on your very nice talk, and asks about, what about total biomass production in those lines with faster PS2 recovery?
That's an excellent question. In terms of time, I removed that slide just before this, but I can briefly give you a taste of it because it's actually really interesting. So from the looks of it, from all the measurements I did, this was destined to be a positive effect. If anything, it would have a positive effect on biomass. And I tested it in 6 different light regimes, and it's both in constant and fluctuating light regimes. And I could see that in most conditions, it did not have an effect on biomass, not negative and not positive. There's one strong outlier to this, and that was when I exposed these plants to highly fluctuating light conditions. So this means that in some conditions, so this was a data set that I collected with in a mature maize canopy. So there was a lot of wind movement, and it had fluctuations up to 10 times per second. And when plants were subjected to such light intensities, actually the positive effect of 5PS2, so this reduction in NADH activity, due to this reduction in NADH activity, had up to 15% lower biomass. So that learned me actually something very interesting, the fact that even though from the outside looking at it, and a trait that can improve photosynthesis is the result of a trade-off that actually is required in some environmental conditions, in this case, highly fluctuating light. So not a clear-cut answer that you probably were searching for, but I think it highlights something very interesting here.
Okay, thank you. Are there any more questions? I've got one as well. Is it possible to attribute an amount of variation to the chloroplast or the mitochondria that's come across? Do you know whether can you Can you make any decision of what's making the biggest contribution to that variation?
Yes. So you're saying basically when you grow plants in somewhere and you say how much of that variation is due to either the chloroplast or the mitochondria, you say. That was your question, right?
Yeah.
So that is effectively what we did, but that's only possible if you have such hybrid panel where you are able to systematically disentangle this. The only thing that I'm not able to do in this way per se, or in theory, is that I'm not able to separate the mitochondria from the chloroplast itself, and that's just because the organelles inherit together. Having said that, using whole genome sequences, I am able to identify accessions that have the same that might not have variation in the mitochondria, but only in the chloroplast, and thereby I can exclude some options. And so, using additional methods, I can still say, okay, it's due to the mitochondria or due to the chloroplast. It requires a bit of extra work, but it can be done.
Okay. And do you ever make hybrids that have got both the maternal and the paternal chloroplasts and mitochondria? So you've got a mix, so you've got the one nuclear.
No. Short question is no. I have not made those, and that's because all of my work relies on crosses, and in crosses there is a very strong. At least in the case of Arabidopsis, the maternal genome is always inherited. And the maternal organelle genomes are always inherited. There are some exceptions, and there's a very low percentage that where via the pollen also organelle genomes are, or the organelles are transferred. But this is such a low percentage that I never systematically studied this. There are some cases where this is possible. In other species, that this is possible because some pines. In pines, the inheritance is different. In cucumber, for instance, so this is different in different species.
Okay.
So, and what you can also do is use protoplast fusion, where you can actually combine these things. So then you fuse them, and then you can select for the ones that you find desirable.
Okay. If there's any more questions, put them in now. If not, we can return to Tom if we've got time at the end for any other questions. So I will say thank you, Tom, and I will move on to introducing Marc. So Marc is part of the Capitalised Consortium, Capitalised project, and is a research associate at IPK, which and he's interested in what's happening in plants at the molecular and phenotypic level under fluctuating light and dynamic environments. He investigates genotype environment interactions under simulated field conditions, including future predicted climates. And today he's going to talk about the system that simulates these climates, which is the IPK phenosphere. Over to you.
So thank you, Louisa. Can you guys hear me and see my slides?
Yeah.
Okay, perfect. So yeah, welcome to my part of the webinar, and I would like to introduce to you the potential of the IPK phenosphere. This is this building here on the right, in which we can field simulate an indoor environment with all the field dynamics. At least that's what we are aiming for, and I will show you now how far we come with that. So what was the incentive to build such a building like the phenosphere? So for example, for research, we saw that there is a need of exposure of plants, especially crop plants, to expose them to relevant field-like conditions, but in a reproducible and adjustable manner, to answer questions like systems biology analysis, to unravel the molecular mechanisms underlying the expression of important agronomical traits, and to further test hypotheses derived from these analyses, for example, modulation of performance-related characteristics in different environmental conditions. Furthermore, I think which is quite important is to study the performance of selected and specifically designed genotypes under different weather conditions, but also including future climate scenarios. So something which we cannot do currently on the field, but I think we could potentially get a head start by using such a system. So what the phenosphere was designed to be, to provide opportunities to overcome the limitations of currently typically climatized glasshouses and climate changes, so in cause of dynamics, amplitude of environmental dynamics, and obviously of size, so something where we can also study populations of crop species. So what is basically the phenosphere? So here you can see a layout, and the image here you can see point of view is coming from the top right angle. So it consists out of 4 large compartments and a processing area where we can process samples and a control area. And these 4 compartments are further separated into 2 systems. So we have once the container system, where we grow plants in large volume soil containers, and a rhizotron system, where we grow plants in rhizotrons and can phenotype root and shoot simultaneously. The container-based phenotyping platform, as mentioned, consists of 2 compartments, are 116 square meters. And so if everything is fully occupied, we can harbor 2 times 54 containers. Each container has an area of 1 square meter, and we can grow plants, crop plants, with an open plant height of up to 4.5 meters, with smaller containers even up to 6 meters. For the rhizotron system, we have again 2 compartments which can be operated independently from each other or combinedly. And we have 2 times 180 rhizotrons available, and an open plant height possible is here limited to 1.20 meter, but this is also limited by basically the root growth. And so we'll first give you a glimpse about the rhizotron system, and then I will talk more about the container system. So the rhizotron system you can see here. So it comprises of these rhizotrons and these imaging towers, which can move on a y-axis and then take out these rhizoboxes. And these rhizoboxes have a dimension of 60 times 19 times 4 centimeters. And their plants are growing with an angle of 42 degrees with a plexiglass side. The plexiglass side is while during the growth is shielding from light. And only when the imaging tower comes, it takes out such rhizoboxes and then phenotypes them in parallel on the vegetative and on the root growth area. And currently, colleagues from the IPK have conducted a huge experiment in the system to assess the root diversity set of a diverse set of species. You can see some examples for example, potato, barley, rapeseed, and also how these phenotypes look like. But this is as much as I want to talk about the rhizotron system. This is another story from the colleagues who actually worked with that. And I would like to focus now on the container-based system in the phenosphere in compartment 1 and 2. So this cultivation infrastructure enables us to assess plant performance in specifically designed and fully controlled environments. This is something which was already true for climate chambers, but I think this system pushes a lot further and allows us now to apply dynamics which are relevant or equal to the dynamics which we find on a field environment. So for example, for elimination, we can increase the elimination to up to 1,500 micromole per square meter per second. And this spectrum is set together by discharge metal halide lamps, UV lamps, and 6 differentially colored LEDs. Furthermore, we can control air and soil temperature from as low as 5 degrees up to 40 degrees, and we do that in very fast changes. Relative air humidity can be controlled at daytimes, go as low as 20% relative air humidity, and during night we go up to 90% if necessary. Furthermore, you can see these black fans, these large fans on both sides. They can also apply a wind simulation with wind speed of 4 on the Beaufort scale, so something like 23 to 28 meters per second if everything is 100% density. The carbon dioxide content can be increased from ambient. I mean, ambient is currently already higher than 400 to 1,200 parts per million, and everything is automatically watered with a drip irrigation system. Furthermore, all the parameters are then monitored with aerial and also soil sensors. So in the end, we have these 2 compartments. So here you can see an image from compartment 1 from the maze validation experiment I'm going to talk about in a minute. And here is a separation wall, and this separation wall can be removed and we can combine. So behind there is another equal size compartment. We combine the space and have a large experiment or operate both of these compartments independently with 2 different experiments, for example. And they were both designed to allow fast changes of environmental conditions and also fluctuating light as they are typical for field conditions. And I think something which is quite nice I would like to show you now are the light sources we are using. So here you can see a photo from compartment 1 where you can see the light panels. So here the big rectangles are the metal halide lamps. So you can see the spectrum of the ceramic metal halide lamps. And then in between we have the UV lamps together with the differently colored LED light bars. So we have white LEDs, deep blue, blue, cyan blue, red, and far red colored LEDs. And we combine them, for example, in the experiments I'm going to show you. We have cloudy, normal, and sunny days, which we simulate. And the cloudy days, we only go for a mixture of LED light bars. And for example, for normal days, we also include the metal halide lamps together with the LEDs. And we can nicely cover the whole range of relevant light spectrum. And if we crank everything to 100%, we can achieve light intensities around 20 centimeters above soil to 1,300 micro-Einstein, and at 2 meter 20 above soil, so for example, relevant for maize canopy, up to 1,500 micro-Einstein. And the variation you can see here was sourced from different positions in the phenosphere. So we measured them for certain container positions, including the borders. So I think we have a nice narrow band of variation here and quite homogeneous light intensity delivery through the system. The containers are actually then the place where we grow the plants, and each container, as mentioned, has 1 square meter of surface area and 1 meter of internal height. So we have basically 1 cubic meter of soil, and the soil is then stacked in a field-like soil structure with several layers. So we start with the top layer. It could be a substrate mixture or a topsoil, whatever the experimenter likes. Then followed by loam or subsoil from the field in the case of the maize experiments, and then followed by 2 layers of coarse sand and coarse gravel. And then there is a fleece and a drainage layer, which is comprised of polystyrol pellets to avoid waterlogging in these containers. And furthermore, it allows us to collect the drained water for further analysis if necessary. All these containers are then supplied with a coil pipe system, which is coiling around the border of the container wall, where we can then put cooled or heated water through to allow soil temperature control as well. So I would like to show you the validation experiment I performed during my PhD thesis to actually back up the claims we are making. So the idea was that there is a suboptimal match of growing condition which hampers the transfer of knowledge, for example, from controlled environments in glasshouses or climate chambers to actually field environments. And so the phenosphere was designed, as I've introduced, to simulate field environments in a reproducible manner, so to basically bridge this gap which we are currently having in systems. And the idea now of the validation experiment was to benchmark that claim and benchmark the effects on plant growth of weather conditions of a single maize growing season and of an average season over 3 years were compared to those of a standard glasshouse and of 4 consecutive years of field trial. And the material we used were 11 maize inbred lines coming from breeding lines and from our gen bank here at the IPK, and they were chosen for their diverse phenology in the vegetative stage, and their origin from North America, Central Europe, ex-Soviet Union countries, and Asia. And here you can see now the temperature profiles, the hourly temperature profiles from early May until mid-September, measured in each environment. So here they are measured just in the outside environment, for the 4 field experiments at the IPK field sites, measured inside the glasshouse, and also measured inside the phenosphere from originating from the simulation we conducted. So you can see that we grow them in double row plots with border planting of a commercially available hybrid. In glasshouse, we grow them in 5.5 liter pots and fertilize and water them regularly. And in the phenosphere, we grow them in these containers, where we can also apply the same double row plot design as we do in the field. And the weather simulation which we conducted, so the phenosphere average was averaging the weather between 2016, 17, and 18 on the field, thereby having the same amplitude, but basically not the patterning anymore, and simulating a single season, trying to replicate the season of 2016 at the IPK, where we also respected not only the amplitude, but also the patterning of how we applied these days in correct order. When you now look at the correlation over the whole season between all these temperature profiles, you can nicely see that the highest correlation was achieved between the field 2016 and its simulation on that. So you can see here that is a correlation with a coefficient of 0.88. Furthermore, we found that the simulating only a single season has a lower correlation than with the other field environments in comparison to the average experiment, where we basically averaged over the 3 years. So we have an overall higher correlation with the other field years, but not as large as we do find for single season simulation. The lowest correlation was found with the glasshouse, with any of the field environments, and also with any of the dynamic environments inside the phenosphere. So, but now we start here and end here. But what is interesting now is how do we process over, how we proceed over time in this system? How do these genotypes respond to the environment we generate and we offer? So when we look here, these are 3 selected genotypes, 2 gene bank lines and 1 B73, like most of you in the maize field know, around 40 days after sowing. And you can see that in the glasshouse at this time, plants are already basically mature. They are flowering this one genotype, and they are tall and slender in their pods and growing quite fastly. In the average between 3 years, we can see that also these same genotypes are already flowering and plants are quite advanced in their development. However, they are more bushy, because they are also subjected to wind treatment and have a more robust phenotype. However, on the field, plants are quite small in comparison to that. When we now look at the plants in the phenosphere simulating the year 2016, we can see that they are basically pretty close to the same developmental phase. When we now look and analyze that effect over the whole population, over the whole time, here the trait plant height, in a nonlinear logistic growth model fitted for the whole population, colored here for each environment. You can see plotted on X is days after sowing and Y is here plant height. Here is first derivative, the growth speed, so basically the plant height percentage contribution per day. You can see that the yellow line, the plants in the glasshouse, as you can already have seen, grow fastest and earliest. Then the average climate is somehow intermediate. And the purple one, which simulates a single season of the year 2016 on the field, the blue line, they're pretty close together. So they basically have no significant differences in when they reach the maximum growth speed, and they track pretty closely. So they are not alike. So likely there are some sources of other variation than just climate, maybe biotic factors, which we cannot control for, but we come pretty close here and can replicate the temporal growth pattern in the simulation in comparison to the plants on the field. So now we further advanced from there. So what we learned is that going with average climate doesn't bring us to where we want to go. So we need to simulate single unique days. Yeah. But we also want to simulate characteristics of a certain area and and integrate data over several years. So for another experiment we're currently running, where we work with a winter oilseed rape population, we chose to base our development of the climate regime on 12 years of historic IPK weather data from 2008 to 2019. When we zoom in, for example, here the year 2016 again, we have nicely hourly resolved value from early January to end, so basically for the whole year. And if we now simulate that, we went for such an approach that we take 1 week, which we try to replicate. So we design our climate regime weekly on a weekly basis. And then we take these 7 days for the week of 21st of October to 30th October for all the 12 years. So basically we have 12 Mondays, 12 Tuesdays, and so on. And then applied a partitioning around Meadow-Eads cluster algorithm on the Manhattan dissimilarity matrix of the hourly resolved temperature profiles. You can see if we add it up, it's only 81 days, so we would expect 84 days. Yeah, even such a sophisticated weather station sometimes has data loss and doesn't record or has issues, so that's why it's quite relevant that we integrate over several years. And what we did, we forced the system to apply us 3 clusters to be able to simulate cloudy, normal, and sunny days. And then we looked here, and we not only determined from these clusters, from these temperature clusters, whether a day is cloudy, but now we also went back to the other data sources, global solar radiation and humidity profiles, to determine whether a day, for example, with the lowest light intensity would be the cloudy day. And you can see that in end of October in Germany, most days are cloudy, some days are normal, and only a few days are sunny at this time point. So we further then try to find as few connector points to replicate these temperature profiles in our system, and weekly adjusted the time point of sunrise and sunset, because in the northern hemisphere we are subjected to season. So if we want to simulate a whole growing season, we have to take care of that, and we did that on a weekly basis. And monthly, we adjust light intensity. So for example, going for highest light intensity, obviously in June and July, and for the lowest in the winter months. Furthermore, I would like to show that we can apply a specific stress treatment in the system. So for example, in the winter oilseed rape project, we wanted to simulate a spring, early summer drought during the flowering time. So what we did was we reduced the watering by mid of March inside the phenosphere for 3 continuous weeks. We replaced the cloudy days by sunny days. So now we only have sunny days and normal days, and then monitored the effect on the plants. So you can see the water content in the containers, and you can see the drought phenotype. So after 3 weeks, plants are properly stressed. And then that was done by Dominik Noch in our research group. He applied a heavy rain event. So heavy rain is something we cannot do automatically, but the system is robust enough that we can go in there with a water hose and spray the plants, and that was what we did. So 3 times a day for 3 consecutive days in a week, for 2 consecutive weeks, we applied rain stress and could induce then after that this pre-harvest sprouting phenotype. So since 1 year, we also have the phenocrane system installed. So the validation experiments I've shown you were all done on manually measurement based. And now we have also the phenocrane system, which is basically a huge crane system, which is supplied with a phenotyping panel consisting of several imaging devices. And we have a phenocrane 1 and a phenocrane 2 in the another compartment. Here you can see the RGB image acquisition. So we have additional light sources on the crane. So we go down to a container and then turn the light on, image, take a capture image. And we can also do that for larger plants. So the crane is height adjustable. Furthermore, we have a 3D laser scanner to assess the plant canopy structure, a fluorochem panel from PSI to measure kinetic chlorophyllescence, for example, to here, in this case, PS2 quantum yield, and furthermore, hyperspectral camera in the visible near infrared range. Here I would like to show you the phenocrane in action. So yeah, this is basically what the system is doing. So we can approach each container independently. And this is any layout is possible. So we're not limited here by a grid structure. So whatever an experimenter wants to do is possible to do so. So you can see just an RGB image acquisition, and then afterwards the crane goes back up and can approach any other position or do whatever the experimenter asks them to do. And I would now also like to show you that during the phase of the experiment, the phenocrane precisely goes to the same position every time. And you can see that even 2 genotypes, so here basically there are 2 different genotypes, we can also assess their development, for example, in flowering time or whatever inside the system. In the capitalized project, we are also going to use the phenosphere. So what currently has been done from the IPK side, we have phenotyped the magic maze panel extensively in our LambdaTech controlled environment phenotyping systems, found QTLs, went for contrasting RILs and immortalized F2s to confirm these QTLs again in LambdaTech. And currently we are working to generating these F2s and will later this year phenotype them in the system again. And then at late 2023, early 2024, we are going to expose these normal immortalized F2s to challenging conditions, because currently in the capitalized project, all these conditions we have phenotyped them under were like controlled, non-challenging conditions. And now we try to see whether these beneficial or detrimental alleles, how they actually behave under more field-like environments where we can treat plants, as I've shown you, to fluctuating and controlled and fluctuating light and temperature dynamics inside the phenosphere system. And with that, I'm already at the summary. So I would say the IPK phenosphere is now a unique plant cultivation facility and is now fully operational. We have the container system where plants are cultivated in stands, simulating field plots of up to 1 square meter, and then 108 large volume containers are available with 1 cubic meter of soil volume in a field-like layered soil structure. It is designed to run carefully planned cultivation scenarios in a highly reproducible manner. So I've shown you that you can basically replicate a growing season, and that the plant growth and performance can now be automatically assessed using the phenocrane system, the 2 phenocrane system in both compartments. And they can also be operated independently from each other. For example, 2 experimenters can run 2 experiments at the same time. Furthermore, we have the other half of the phenosphere is basically the rhizotron system, where we can now phenotype root and shoot traits simultaneously in 360 large rhizoboxes to assess variation in root growth and developmental dynamics. And I would like to acknowledge all the scientists involved, but also, which is quite important, to thank all the gardeners, technicians, engineers, and bioinformaticians in the background who do all the work. With only scientists, you wouldn't be able to stem these projects. Obviously, I would like to thank all the funding agencies, and I would like to point you, there's the option on our website to go to IPK-Gattersleben.de Institute Locations Digital Tour, and then get more insight about our facilities. And I would like to thank you for your attention.
Thank you, Mark. Does anybody have any questions? If so, please put them into the question box, and I will put them to Mark. But I will kick off. First of all, I'd like to say there must be a lot of people out there with greenhouse envy. I know I certainly have. If anybody who's done any experiments in a glasshouse and you're trying to control various things, you're never managing to control this many. If you were to try and control some of them, which would you say were the most important factors? If I was trying to control, for example, if I was running a trial, would it be trying to simulate the lights or adding the wind factor if I was trying to make it more realistic? Is there any factors that are more important than others, would you say?
I think this is something which I hope we find out if we do this experiment where we now have like the basis. Yeah, we can replicate field-like conditions, and now we can play around with singular factors to see which is it's the largest factor. But what I can tell you from personal experience as an anecdote, so I would say, especially for maize, this wind is quite important to have the plants invest also in the stem, because otherwise they just grow and you have to fixate them on sticks for them to not bend over. But obviously
how do you make that? Yeah, is that sort of controlled in a simulation way, or is it wind put in different directions, as in?
So the system here, you can see we have two opposing ventilators on both sides, and we can operate them in push-pull configuration. So we can change the wind direction. And so what we are doing, we apply always wind. So even during night times on the field, we have a mild breeze, which is still there. So even in the nighttime, they are operating. And during daytime, they have peaks of wind, as we find normally for standard days. So we can't simulate thunderstorms or anything like that, or extreme events, but we can apply significant wind effect. And so on the IPK field side, luckily, so we also have a wind directometer, or how this is called, and we know that the wind, the IPK, comes from two opposing directions. So it's perfectly designed, well, pretty easy to simulate two opposing directions. So it would not be possible to simulate like rotating wind or something. Yeah, this is not possible. This is a limitation of the system. Yeah. But we can pulse them and we can change direction. That's what we are doing. Yeah.
Yeah, nice. And in terms of simulating climate change conditions, I mean, we have, certainly in the UK recently, we've had some very frosty, well, we might have some very spring days where it's very warm, and then we'll have a few days of really frosty conditions. But you couldn't simulate that, could you? Because you are 5 degrees. So is that something they might, is that something that...
We can use the outside temperature. Yeah, so if you're like doing an experiment in the winter, we can use, for example, cold air from outside, because I mean this uses a lot of energy to cool such a system. But the lower limit is 0 degrees Celsius. Otherwise, our systems would freeze, and that's basically the physical limit of the system.
What's your upper limit that you do?
Upper limit is more than 40 degrees Celsius.
Oh, wow.
Yeah. Yeah, so I think the upper limit would then be the cooling of the lamps, yeah, of the metal halide lamps, but yeah, and the LEDs in the end. But they have an additional cooling system, so yeah, we can go higher than 40 degrees Celsius, and basically simulate very, very dry and hot weather inside there if we want to. Yeah.
So you talk about having data you've collected from close to the IPK site, haven't you, to simulate? So are you collecting data from anywhere else, like, I don't know, southern Spain, that is really potentially more extreme than what you're getting there?
So for these validation experiments, we used 4 years of IPK field data, which I as well phenotyped on the IPK field grounds. But for example, for the Avatars project, where we're doing the winter oilseed rape experiments, we work with the NPZI, and they give us seeds and phenotypes from their different location of field experiments. We currently have the Dromamate project, which works with maize, but with Mediterranean germplasm. So yeah, there we have also, I think, phenotypes as a comparison from Spain, or I think even from Northern Africa. So yeah, that would be possible. In the end, I mean, yes, the limits are 0 degrees Celsius, so we can't go lower, we can't simulate frost, but any of the warmer climate should be no problem. And typical crop growing season in Europe should be no problem as well.
I think it's an amazing system. I think, you know, even like salinity, and presumably you could do waterlogging experiments if you wanted as well for all of those to phenotype.
Sure. We could plug the holes in the container and then do flooding if we want to. And I think we can also, if Tom manages to have these hybrids in crops, I mean, we could also now have a system where we can have fluctuating light in a very fast manner and expose basically crop populations to that, and use the FluoChem device to do these experiments. So quite exciting what we could do in the future. And
you're not limited to large plants, are you? You can do small.
No. So we have, as I said, basically 4.5 meters, or from the graded floor up to 6 to 8 meters of isothermal space where we can grow the plants. So we have a mild increase in light intensity. The closer we come to the light sources, but as I've shown you, this is minuscule. So 4-meter plant height would be no problem.
Yeah. Is there any more questions for either Marc or Tom? Write them into the chat now. I'll just check there isn't any hands up. Don't see any hands up. I think, do I have any more questions? Oh, I do have a question. And this is for a personal, having set up trials myself. So are you changing the soil in these 1 meter cubed containers for every experiment?
Depends. So we did experiments where we tested on maize on 2 genotypes. So I tested, was one additional experiment I didn't show, that was just testing the homogeneity of the system. And I also, for some containers, I exchanged the whole soil, just the topsoil. Basically, at least for one, so in this container, 1 generation of maize was grown prior. The soil had nearly none effect. So we tested like total variance decomposition, and basically the effect was like sub 1 or 2%. But this is in the end done by the experimenter. So for new experiment series, we exchange everything. Yeah. So this is quite. That's why I said we need all these gardeners and engineers. So yeah, to setting up such an experiment and to controlling it, maintaining it, I think this is the biggest challenge here. And yes, we are doing that. And for sometimes we only exchange topsoil level and replace only the first 25 centimeters. This is also feasible, for example, from one maize to another maize generation. Yeah, It's a lot of work.
Well, I can't see any more questions in the question box, so I'm gonna thank both our speakers for presenting such really interesting talks. And just to remind you that the next webinar is on the 2nd of May with Thomas Naegeli. He's talking about stabilizing photosynthesis in a dynamic environment, and about a trade-off with photorespiration. So I think it should be an interesting talk. Please don't forget to register. Thank you all for attending. Thank our speakers, and I look forward to seeing you all next month.
Thank you, Louisa. Bye.
You're welcome. Thank you. Bye-bye.